Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. All right, before we begin, are there any questions? Go to chapter 1. I thought I opened that. Yeah, I did. There it is. All right, let me share my screen. Let me add that I did some grading. Lab 00 is graded, and I'm about halfway, maybe a little bit further in Lab 01. I hope to have Lab 01 finished tonight. If I do finish it tonight, I will then send out, an announcement with a comment with your grade in the assignment stating that you have until this Saturday to turn in Lab 00 and Lab 01 for the second time, which you need to do if you did not get 100% the first time, meaning in the lab you have 2 attempts to get 100% in the lab. Any questions about any of that? I have not put that announcement on Lab 01 because I was trying to get Lab 0, excuse me, I haven't put it on Lab 00 yet because I was trying to get Lab 01 graded and I was hoping to get it all graded before this class began, but I did not succeed. Any questions about anything? All right, let's go to the syllabus. Today is, October 1st. And we're supposed to be finishing chapter 1 and starting chapter 2. Let me warn you that this Wednesday we have our first practice quiz. It's going to be over chapter 1 and lab 0 and lab 1 and the syllabus. Any questions about any of that? You will get credit for taking the practice quiz. It says it's out of 0, but that's because it's extra credit I'm going to give you. And I think it will be 1 or 2 points. I'll have to look up. I don't remember if it's 1 or 2 points, but something like that. Of course, that'll depend on how many answers you get correct. I think on the practice quiz, you can take it twice. So if you don't get 100%, you can take it again to up your grade. Any question about any of that? All right. Now I have that we were here in the lab. Can somebody confirm that we discussed what microbiology and microorganisms were and what falls under the umbrella of being a microorganism? Is this the last thing we talked about? Okay. Let me briefly talk about viruses. Viruses are acellular. They do consist of a genome of either DNA or RNA, which is their core molecule. The core is surrounded by a protein coat, and in some viruses, that protein coat may be enclosed inside a lipid envelope. Not all viruses have an envelope. And when do viruses replicate? They only replicate when they are in their host cell. They are not part of the three domain system because they do not have cells. They're really not an organism. Any question about that? All right. We'll talk about viruses much later in the term, but I did need to talk just a little bit about viruses there. When we compare the three domains of cellular organisms, bacteria, the archaea, and the eukaryotes, we note that the cell type for the bacteria and the archaea is the same. It's cell type prokaryote, and for the eukaryotes, the cell type is eukaryote. When we look at the chromosome, bacteria and archaea generally have one chromosome, which is in a circle, meaning the ending place is the place the chromosome began. That differs from eukaryotes that have many chromosomes, and there are linear chromosomes. Like if we look at the X chromosome, it'll have a beginning spot here and an ending spot there. And those two they're not the same. We can also look at bacteria and the eukaryotes, and the eukaryotes have histones in their chromosome. The DNA wraps around this histone protein, and the bacteria lack histones. Any question about any of that? Furthermore, when we look at the ribosomes, the bacteria and the archaea have smaller ribosomes than the eukaryotes. The eukaryotes have something that we call 80S, and the archaea and the bacteria have smaller ribosomes that we call 70S. The S stands for the sedimentation coefficient. It's the way that biochemists and microbiologists measure ribosomes where they sediment on an ultracentrifuge gradient. And you really don't need to know that. You only need to know that the bacteria and the archaea have slightly smaller ribosomes than eukaryotes, and they're 70S, and the eukaryote size is 80X. Furthermore, when we look at the presence of peptidoglycan in the cell wall, we find it in the bacteria. We do not find it in the archaea or in the eukaryotes. Any question about any of that? All right. So now we're going to change gear and we're going to talk a little bit about the brief history of microbiology. The history of microbiology, especially early, was concerned about the debate over whether spontaneous generation um, could give rise to microbes or whether microbes had to come from another microbe. And the early history of microbiology is also called the golden age of microbiology. You don't need to know the time frame, but 1857 to about 1914, where whenever microbiologists looked at something, they discovered something new. The golden age of microbiology. So early on in microbiology, there was a big debate of whether microbes could spontaneously generate or whether they had to come from other microbes. There was never a question about where a baby came from. People always argued that babies came from their two parents, but there was debate as to whether organisms could spontaneously generate. And we'll talk a little bit about that. And there's a brief history of microbiology showing it in one slide. We'll go through largely the golden age period right here and a few things after that. In the history of microbiology, it actually begins in ancient times. The oldest part of the history of microbiology is you can find fossil record to the ancestors of bacteria today, meaning the first life we have on the planet was microbes in the fossil record, which you can find at 3.5 billion years ago or older. And then the next important, I guess, date in the history of microbiology, there's lots of other dates, but the next important one was in 4300 BC, the Babylonians wrote clay tablets about how to make beer. So they had recipes for making beer. And they didn't know that microbes were involved in making beer because they couldn't see them. But you do need microbes to make beer. And so that's an important date in history of microbiology. Let me state, I'm going to give you the dates. However, you don't need to know any of these dates in the history of microbiology, except for this first date. You should know that the first fossil record for microbes came from 3.5 billion years ago or earlier. And then the next date, 600 AD, the Mayans have written records for making a fermented beverage from cacao, which we would call chocolate today. And there's an actual fossil from 3.5 billion years ago. That's the artist's rendition of it. The next date is 1590. You don't need to know the date. I'm just giving it for a time frame. And that is Janssen developed the compound microscope. And then in 1665, once again, you don't need to know the date, Robert Hooke reported that living things were composed of little boxes that he called cells. And all of these dates I gave you, except for maybe the very first, are before microbiology began. We needed to have the development of the microscope before we could see microbes. And then we had to have the first, I don't know, picture of a cell observed, which really was from a plant. It was not from a microbe. That's why it's before microbiology. Robert Hooke, let me go ahead and show you what he did. If I can find paint, there it is. What he did was he took the bark of the cork tree, which is actually called cork, and you can also use that to make the cork in the bottle of wine. So these are three different words of cork. The tree, which he studied, and he took the bark of it and took thin shavings of it and looked at it under his microscope. And what he observed were boxes like this, slightly beyond where I should have done there, but it's all right. I'm not a great artist. And this reminded him of cells in a monastery. So let me get the eraser here, get that out of here. So we'd have a monastery, and then this would be the building here. Let's draw that there. Oops. And this would be the pathway or hallway. And then these would be the little rooms that the monks did their stuff in, probably slept in and other things. I don't know, maybe read, prayed, putting in a door there. I'm not going to put many of them in. And they called these cells. And he was reminded of these rooms in the monastery, and that's what it looked like under the uh, microscope. And see, so he called them cells after the rooms in a monastery. Any question about that? And that's what we call them today, cells, the basic organization of all life form. Now, when Dr. Hooke looked at plants, he found cells under the microscope. But when he looked at animal tissue, he could not see cells under the microscope. And the reason was is that the microscopes in his days could not focus very well, and the cells of plants could be seen with the poor focusing of the microscopes, and the cells of animals just could not be seen because the microscopes couldn't focus in well enough to see the cells. What is different between a plant cell and an animal cell that would allow a plant cell to be seen under a blurry early microscope? What's the difference between a plant cell and an animal cell?
A cell wall.
Yeah, plants have a thick cell wall, and that allowed Robert Hooke to see the cell under the plant, trying to make that nice and thick. And then animal cells did have a cell membrane, but that's very thin. And so when Robert Hooke looked at the tissue, he was not able to see the cell membrane, but he was able to see the cell wall under plants. And that's why he saw that plants had cells. It took a microbiologist 100 years to improve the microscope after Robert Hooke looked before people could use a microscope and then see cells in animal tissue. And it just needed the advancement and improvement of the microscope where they could focus well enough to see the cell membrane on an animal cell. That did mark the beginning of the cell theory that all living things are composed of cells. And this is so common today, we don't even really refer to it much as a cell theory. But this was developed further by others, and Virchow actually published it and wrote about it in much after 1665, 100 years or later, he did that. That all living things are composed of cells. All right. So in the history of microbiology, there were two hypotheses to explain how microbes originated, and there were competing hypotheses. One was that microbes arose by spontaneous generation. The theory was a vital force came in to matter, usually organic matter, and then spontaneously generated a living life form. And the other theory is that all life came from pre-existing life, and that was the theory of biogenesis. Over a century ago, there were beliefs like if you threw a log in the fire, sometimes a newt would crawl out of the fire. And people would say, spontaneous generation of newt. Wood and fire spontaneously generated a newt. In reality, the newt was probably in the wood, like in the bark of the wood. And then when it was thrown into the fire, it just was escaping from the fire to avoid the heat. But people noticed flies emerging from manure, and they would say, spontaneous generation of flies. The vital force came into the manure, giving rise to the flies. And the same with maggots. In a decaying corpse, maggots would arise in the corpse, and people say spontaneous generation of maggots. Most were logical, not any were scientific, and some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar that had wheat kernels in the bottom of the jar, it would spontaneously give rise to the mice. In reality, the mice climbed into the jar to get the wheat kernel to eat them, and they simply got stuck in the jar. They weren't able to climb out. The sweaty underwear had nothing to do with the spontaneous generation of the mice. Brief sidetrack, and that is if you have a tube with media in it, and then you let that sit, it would become turbid with microbial growth. And this was seen all the time early on when people were working in microbiology. And that's why people were wondering if microbes could spontaneously generate, because you had no microbes. And then later on, you had a team full of microbes in the tube. Um, Let me go back to here and state that what happened was the tube was not sterile, so there were a few microbes in it. And if it wasn't capped or wasn't capped right after people put the media in, that the microbes would also fall down from the air, and then microbes would grow into the tube until the tube became cloudy. And that we call turbid, by the way. Cloudy, the media turning cloudy. The first evidence uh, to answer the question of whether life spontaneously generates or not came from Francesco Redi. And this guy you should know because not only did he evidence for the first, well, evidence against spontaneous generation, I'll word it that way, but he also was the first person to set up a controlled scientific experiment and then write about it and publish it. In 1668, once again, you don't need to know the year, but you should know Francesco Redi set up an experiment where he had an open jar. He put meat in the jar, left it open, and then maggots appeared in the meat. He had three completely sealed jars, putting meat in before he sealed it, and then he noticed no maggots in the meat. And the people who believed in spontaneous generation said, no, that doesn't work. You sealed the jar, you prevented the vital force from coming in, and then spontaneously generating the maggots. So he ingeniously got three jars, put the meat in, and then covered them with a fine wire net. It was a metal wire net so that the vital force could come in, but the meat did not give rise to maggots. He didn't know this at the time, but the maggots were coming because flies were flying in the open jar and laying eggs in the meat, giving rise to the maggots. And the flies could not get in the completely sealed jar or the jars with the fine wire net, mesh netting. Anyways, his results did not support spontaneous generation. After his time, nobody believed that complex organisms, we're talking about eukaryote animals, maybe plants as well, did not arise by spontaneous generation. However, people still believe that microbes could spontaneously generate. And that's because the microbes could get through the wire netting and then grow in the meat, for example. Francesco Redi didn't look at microbes, but about this time was the first observations in microbiology that happened by Antony van Leeuwenhoek. I'm not Dutch, so I could be mispronouncing his name, but he described seeing living microorganisms in everywhere he looked. in scrapings from his teeth, in rainwater, in peppercorn infusions. And then he looked at the water from the River Thames, which the people in London drank, and he found tons of microbes in it. This is actually his one of his microscopes. It is a simple microscope because it only has one magnification lens, as opposed to where was it? Robert Hooke's compound microscopes. However, because he had only one lens, he was able to see clearer with his microscope than people were with a compound microscope. And then he was able to focus in on microbes. It's too bad he didn't look at animal cells because he probably could have been able to see the cells in animal tissue, but he didn't look there. He was looking at microbes. And not only did he see all sorts of yeast cells and fungi and protists, trying to think of some algae, things like that, but he was able to see cells so small that they were not seen by other microbiologists for nearly a century later. And that is, he saw cells here that you can recognize that these are bacteria. That's sorry, I'm drawing a blank here. A flagella? No, not a flagella. These are cocci right there. Let me blow that up. There we go. Cocci right here. These are bacilli. That's what I'm trying to say. That's the movement of bacilla. There's a spirillum that he drew. And then down here are various bacilli. That's either a bent bacilli or two cells stuck together. The point is, he saw bacteria, cells that were so small they were not seen again for 100 years. And that's just because they needed 100 years for the microscopes to improve before people could use them and see bacteria under the compound microscopes. Uh, Antony van Leeuwenhoek was the best microscope builder of his time, and then the best lens grinder of his time. He did everything, um, looking at the cells, making his own microscopes, and then grinding his own lenses. He was the best lens grinder of his time. And that's why I was able to see things that other people could not, including microbes. Any question about that? All right, you don't need to know the year, but in 1858, Rudolf Virchow proposed an extension of the cell theory that said living cells can only arise from pre-existing living cells. This became known as the concept of biogenesis, and he argued that all microbes came from other microbes. However, the argument about whether spontaneous generation or not continued for 3 more years after he stated his theory and wrote it up. And it wasn't until 1861 that spontaneous generation was finally put to rest by none other than Louis Pasteur. And he disproves the theory of spontaneous generation, thereby confirming the theory of biogenesis. What Louis Pasteur did was, first, he showed that microbes are present in the air and that they can fall out of the air and then cause contamination of media. But he also showed that microbes can be destroyed by heat And then he made a famous, ingenious swan neck flash experiment where what he did was he took a glass cylinder, put media in it, and had a long glass neck, and then he heated the neck to get the swan neck like this, and then he heated the media to sterilize it to kill the microbes that were in the media and in the container. And then he left it open to the air so the vital force could come in and mix with the media to spontaneously generate the microbes. And he saw no microbial growth in the media. He didn't see any microbial growth in the media unless he brought the media down to here and then put it back in the flask. And what he proposed was that microbes were falling out of the air and then would fall down the glass tubing, and then they would accumulate here. They would not go up against gravity, so the microbes would be there. And that's why the media would not have any microbes in it if he put it in the swan neck flask and treated the flask like this. And you can actually go outside of the microbiology lab. I haven't looked for, gosh, I guess probably since before COVID, but there was one of these in the window of the microbiology lab that was set up probably about 15 years ago, and it still has no growth in the media. Any question about any of that? Anyways, after Louis Pasteur's day, we now state that microbes only arise from pre-existing microbes. The beginning of Pasteur's work discovered many other things, including the relationship between microbes and disease. This we call the germ theory of disease. We also came out of Louis Pasteur's work the concept of immunity and the concept of antimicrobial drugs, that you could find a drug which works against a microbe. Shortly thereafter, Pasteur developed the basic idea of aseptic technique, where you keep the culture under aseptic conditions, and then it doesn't get contaminated. And so if you have a pure culture, it'll remain pure. Koch developed, and then he also, I don't have that up here, but he developed a technique for getting a pure culture. It was very laborious. He simply used limiting plating dilution. He was using a liquid broth, and he would make different dilutions. And if only one replica out of 10 of a certain dilution got growth in it, he correctly concluded that one or a few cells gave rise to that growth, and so that culture was most of the time pure. And you had to have, like I said, 10 tubes. 9 of them would have no growth because so few microbes were put into it that 9 of them got none. And then 1 out of 10 would get microbes because one or a few cells were put into that tube, and it would then grow up. And most of the time it would be a pure culture because it would be most of the time would be started by one cell. Excuse me, not Louis. That was a difficult way to get a pure culture. Dr. Koch developed a much easier way of getting a pure culture, and that is he did streaking, colony streaking, on a solid media. And if you streak it out 2 times through 2 colony formation events, you can then say that you have a pure culture. You should know if you streak it out only 1 time, only about 70% of the colonies are pure. So you do need to streak it out through 2 colony formation events. Any question about that? About the same time, Hess, a person working in Dr. Koch's lab, developed a nutrient auger. It was a new solid medium for growing lots of microbial growth. And I think we talked about nutrient auger. It's one of the most commonly used media in microbiology today. And about this time also is when Winogradsky developed the concept that microorganisms were involved in the biogeochemical cycles, such as the nitrogen cycle and the sulfur cycle. And as I mentioned, Winogradsky was able to discover this because he, unlike almost all other microbiologists, did not work with pure cultures. Winogradsky was working with a community of microorganisms, and you need a community of microorganisms to see the biogeochemical cycles, such as the nitrogen cycle. All right. After Pasteur showed that spontaneous generation does not give rise to microbes, Louis Pasteur then went on to show that microbes are responsible for fermentation, meaning microbes are responsible for making beer and wine, as well as leavened bread. And he showed that alcoholic fermentation was the conversion of sugar to alcohol, which is needed in the making of beer and wine. He also went and showed that microbial growth is also responsible for the spoilage of beer and wine, and that it was another microbe going in and converting the alcohol to acetic acid to make vinegar. So other microbes such as bacteria converted the alcohol, like in the beer and the wine, to vinegar. With Louis Pasteur's work on fermentation and spontaneous generation, people then began wondering, could spoilage bacteria, sorry, I'm not there yet. Let me go ahead and go with that train of thought. Could that realization that microbes can affect organic matter, making fermentation, could microbes have a similar relationship with plants and animals? Might microorganisms cause disease? This concept became known as the germ theory of disease. The different germs cause disease. This was an important concept because originally people believed that poisonous vapors were responsible for causing an infectious disease. Like people would say, you went through the swamp, you breathed in the poisonous vapors of the swamp, and then you got cholera. That's what people stated before the germ theory of disease. Of course, now we know in the germ theory of disease, the cholera bacteria happened to be in the swamp, and somebody go walking through the swamp could breathe them in and then develop cholera. Any question about any of that? Louis Pasteur, with his work on fermentation, discovered that heat could be applied to kill microbes, and that if you used heat that was fairly warm, you did kill many of the, if not all of the spoilage bacteria, but would not evaporate all of the alcohol in the wine. Meaning you would evaporate some, but most of it would remain. And it would be warm enough to prevent the spoilage bacteria. You'd kill the spoilage bacteria in the beer and the wine. He called this pasteurization, the application of a high heat for a short period of time to pasteurize a wide variety of foods. He initially used it to pasteurize beer and wine, but today it's mostly used in pasteurization of milk. The milk is not sterile. It does have a shelf life, but its shelf life is extended because it's pasteurized and it has much fewer organisms in it, including no human pathogens, once it's been pasteurized. But there are some microbes that can survive pasteurization. And that's why pasteurized milk, even in the refrigerator, has a shelf life of about 2 weeks. Beyond that, the bacteria that's in the milk could spoil it. Any question about any of that? The germ theory of disease was then advanced by Ignaz Semmelweis, and I'm not German or Austrian, so I'm probably butchering that name, and Joseph Lister, who were working in the hospital and noticed that simply the washing of hands reduced the incidence of disease in the hospital, as well as using a chemical disinfectant to prevent surgical wound on surgery patients. And then the germ theory of disease was literally proved by Robert Koch, who showed that the bacterium Bacillus anthracis causes the disease anthrax. He provided the experimental steps required to prove this, and he wrote these steps up, and today we call those steps Koch's postulates, the steps to prove that a given organism causes a given disease. And what he did was prove the germ theory of disease. He didn't propose it, he proved it. And that's because he showed that Bacillus anthracis causes the disease anthrax. Any question about any of that? Let me shut down my email because I'm getting noise when an email comes through. Oops, sorry. Not supposed to be seeing that. We'll talk about Koch's postulate at 3 times. This is the first time in this term. So obviously it's an important concept. We're going to come back to it and discuss it two additional times. Around this time, we had the first vaccine. Edward Jenner did the first vaccination. He vaccinated people against the disease smallpox with a vaccine that prevented them from getting the disease smallpox. How he did that was he inoculated a person with the cowpox virus, and then he noted that the people inoculated with cowpox were immune to smallpox. It was not understood how his vaccine worked, and it was not understood until the work by Louis Pasteur, where Louis Pasteur showed that the cowpox virus is related to the smallpox virus. And when you vaccinate someone with cowpox, the person's immune system responds to cowpox and then attacks the cowpox virus. And some of the immune response will also attack the smallpox virus because the two viruses are similar. And that cross reaction gave the immunity to smallpox from Edward Jenner's vaccine. The term vaccination is derived from the word vacca, which is a word meaning cow. So vaccine comes out of the work that Edward Jenner used using the cowpox virus, a virus causing a disease in cow, and the protection is called immunity. Let me state that Edward Jenner really never showed that the cowpox virus protected patients against smallpox. He just believed it because milkmaids who milk the cows tend to pick up cowpox, and they did not get the disease smallpox once they had cowpox. And he was using the cowpox vaccine before he proved it. Obviously, once he used it and then showed that the people vaccinated with cowpox didn't get smallpox, um, he then was able to prove it. But initially, he didn't have anything other than his beliefs. And he first experimented on children. Um, was it Joseph Lister, the first boy he experimented on? And then his own son, the second boy he experimented on. And he was able to experiment on children because back then nobody cared about children. Any question about that? I'm being a little facetious, but Edward Jenner was experimenting on children, and he didn't really prove that his vaccine would work against smallpox. He did show it later when the children were alive and never got smallpox, when smallpox, what do you call it, an epidemic came through, and the two children he vaccinated were immune to smallpox. Unfortunately, both of the children died in childhood of some other illness, but it was not because of smallpox. And back then, there was just a very high child mortality because there were no antibiotics. And if a child got a disease, there wasn't a whole lot you could do to cure the child of the disease. Just needed the child's own immune system to throw off the disease. And both of the children got sick and then died of their illness, but not from smallpox. Chemotherapy was the treatment of disease with chemicals, and this came out of Louis Pasteur's work in the germ theory of disease, that if disease was caused by germs, maybe we could find a chemical agent that could cure disease. And initially, chemotherapeutic agents was and chemotherapy was the term used to for drugs that we used against infectious disease. However, nowadays chemotherapy is used simply as a drug to treat cancer, and we don't call a chemotherapeutic agent something that we use against an infectious disease. We call it an antibacterial or an antimicrobial agent to prevent infectious disease. But chemotherapeutic agent can be used against an infectious disease. An antibiotic is the term we use to use if it's a drug that can work against a bacterial agent. Antibiotic, with the textbook definition, is a chemical produced by a living organism that can kill other microbes. So penicillin G is a chemical made by the penicillium mold, penicillium mold, penicillium mold, and it is a antibiotic. By the textbook definition, oh gosh, ampicillin and carboxylicin are not antibiotics because they are not directly produced by a living organism. Um, Ampicillin is made by a living organism, but then it's chemically modified by a person in a lab to make it ampicillin. And then carboxylicin is man-made. So it is not an antibiotic by the textbook definition. What we'd call it would be a semi-synthetic antibacterial agent, meaning a man-made agent. I won't use the term antibiotic that way because most Americans, including most clinicians, use the term antibiotic against all microbes that cause, not all microbes, all bacterial microbes, meaning all bacteria that can cause an infectious disease. And so that's the way I'll use the term antibiotics. But you should know that there is a textbook definition as well as a common usage of the word antibiotics. Any question about any of that? All right. The first chemotherapeutic agent that we ever came up with to treat a infectious disease, which was actually quinine from the bark of the cinchona or the cinchona tree, both pronunciations are correct, which was used to treat malaria. And Paul Ehrlich, looking at different chemicals, I think it was like his 505th chemical that he looked at, actually found a chemical that could be used as a drug to treat the disease syphilis. And he discovered this drug in 1910. And he called it a magic bullet, a drug that could work against a microbe to cure disease, and then left the host cells relatively unharmed. Why he called it a magic bullet. But the first drug, quinine, from the bark of the cinchona tree, was not found in 1910. Does anyone want to know? Anyone want to guess when they think the first drug was discovered in Europe? Anyone have a guess for a year? Just take a guess.
1950.
1950? No, that would be when about the first penicillin was developed clinically. Actually, World War II. So in the 1940s is when the first antibiotic was made. So when was the first drug against malaria, quinine, discovered? In Europe. Anyone have a guess? Just guess. Somebody just guess. One more guess.
The 1800s, I don't know, somewhere in there.
There, that's a good guess. No, it's actually the mid 1600s. What happened was Jesuit missionaries in Peru noticed that the natives of Peru would eat the bark of the cinchona tree to treat the malaria. And they noticed that it worked. And so they sent the bark of the cinchona tree back to Europe in the mid 1600s. A long time ago, is my point. And who knows when the natives of Peru first discovered it, because they didn't have written records. But obviously before the mid 1600s. Any question about any of that? 1910, reminding you, you don't need to know the year, but that's when Dr. Ehrlich, discovered the first drug for curing syphilis. And in 1928, Alexander Fleming discovered the first antibiotic. What he did was he streaked out a plate bacteria, and he was a lousy microbiologist and got contamination on the plate. And then he left the plate to grow at room temperature, came back over the weekend. Then he just left it on the counter and put it instead of putting it in the incubator. And both the bacteria grew as well as the mold. And Alexander Fleming noticed a ring of inhibition around the mold where the bacteria could not grow. And he correctly concluded that something was being made by the mold, the penicillium, and migrating through the agar, preventing the growth of the bacteria. And he called this penicillin G, actually, the first antibiotic discovered. He was not able to isolate penicillin because the technique were not available to him, but he did experiment with the antibiotic. Couldn't treat it patients because he couldn't get anything other than media that he was growing the mold in, and the media had mold spores in it, so you can give that to a patient in any way either. The first antibiotic that clinically became available was sulfonamide, one of the sulfa drugs, which was a man-made drug, and it was man-made and then purified, so easily purified, and became clinically available in 1935. Penicillin first became clinically available in the early 1940s, so World War II. So it was the second antibiotic that became clinically available, the first one discovered. You should know the bacteria once classified as plants, hence the term flora or microflora for describing microbes. It is the term that actually I was taught when I went through microbiology, microflora. And if you hear me say that, it's just because I forgot to update it. Will you now use it microbiota to discuss microbes growing in and on a region? The normal microbiota are the microbes that are normally present in and on a person or an animal or a plant. And we have the human normal microbiota, and then the kitty cat microbiota, normal microbiota, and they're not the same. Many of the organisms would be, but like on the kitty cat, they have Bartonella henselae, part of their normal microbiota that humans do not have. And if humans ever have Bartonella henselae growing in and on them, it will cause cat scratch fever, a microorganism that causes a disease. Any question about any of that? Let me state that all animals and plants host a symbiotic microbial community, and that they're actually central to the welfare of these animals and plants. The normal microbiota is normally found in and on the skin, the oral nasal cavities, the respiratory tract, the digestive tract, and the urethra, particularly the lower urethra. It's usually not found internally, like in internal organs and blood, or internal tissues, like the liver. The normal microbiota can prevent the growth of pathogens. The normal microbiota can compete for space and resources. The normal microbiota can actually secrete toxins that inhibit the growth of pathogens. We'll talk a little bit about that later. The normal microbiota can also provide nutrients and growth factors for their host. For example, humans get a significant proportion of their folic acid and vitamin K from their normal microbiota. The microbes in our gut make a significant portion of our vitamin K and folic acid. And then lastly, the normal microbiota are important in helping us digest our food. I think everybody knows that E. coli, as well as many other normal gut microbiota, can help us digest our food. The normal microbiota do not normally cause disease. However, they can cause disease in circumstances, such as when they're growing where they should not be growing, and when they are growing in a patient who's immunocompromised, like an AIDS patient. If an infectious organism causes a disease, an infectious disease, then we call it a pathogenic microbe. If the pathogen overcomes the host resistance, we will then have an infectious disease. Any question about any of that? There is a concept of an emerging infectious disease. This is a new or changing disease that is increasing or has the potential to increase in incidence. Can anyone name an emerging infectious disease that we have today?
Influenza?
No, influenza has been around from ancient times for as long as we have written records, and so it is not an emerging infectious disease. Now, if you say the bird flu, that is a new and emerging infectious disease. It is a disease that's been around birds for a long time, but only recently have we discovered that it can cross from birds to humans. And usually you need to have a person who's got very close contact and a lot of contact with birds before they pick up the bird flu. Fortunately, the bird flu does not spread person to person. It does spread bird to bird, and that's because it spreads from the fecal to oral route, meaning the bird poops it out, and then another bird will eat something on the ground, picking up the poop, and then get it orally. And it does spread bird to bird, but fortunately, it doesn't really spread person to person. A person almost always has to get it from a bird. So bird flu is one flu that is an emerging infectious disease, but other flus are not. What's another emerging infectious disease? Something that we're currently under a pandemic.
COVID-19?
COVID-19. It is still considered new and therefore would be an emerging infectious disease. Eventually, COVID-19 will leave the new category, in which case it's not an emerging infectious disease. Like I think HIV causing AIDS has now been around long enough that it is not considered an emerging infectious disease anymore. I could be wrong, but I think it's now been around long enough that it's no longer called an emerging infectious disease. But let me just check on that.
I have a question.
Uh-huh.
So there's been an outbreak of Bordetella pertussis. Would that be considered the emerging infectious disease also?
You're saying an outbreak of pertussis?
Yeah, Bordetella pertussis.
Yeah, no, that would be a disease known once again to humans from ancient times. We have a current outbreak of it, but that would not be a new and emerging infectious disease. That would be a Um, all right, I was wrong here. AIDS is still considered a new and emerging infectious disease, even though it's been around since the 1980s that we know of. It's still considered fairly new and is not a new and emerging infectious. It is an emerging infectious disease. So just to let you know, I was wrong there. I'm not sure when the time span is when they no longer call it a new emerging infectious disease, but actually, gosh, that's almost 40 years ago. All right. You do need to know that what an emerging infectious disease is, a new or changing disease that is increasing or has the potential to increase in incidence. And the concern with bird flu is it might infect people and then change and then spread person to person. So far, that has not happened, and hopefully it'll never happen, but the concern is that might happen. All right, I've got other slides talking about emerging infectious diseases. This is just for your own benefit. I'm not going to quiz you on it, and I'm not going to discuss it, other than you do need to know what emerging infectious disease is. I'll briefly state that West Nile encephalitis is one that recently hit the United States. I don't remember if that was the 1980s or the 1990s. It was not, West Nile virus was not in the Western Hemisphere, meaning the Americas, until that point. And it initially was brought to this country by somebody in New York City. And what they should have done was, what do you call that? Bomb the whole city with insecticide to kill all the mosquitoes. And that would have gotten rid of the West Nile virus. What they did was nothing, and except maybe limited spraying, and it did not control the West Nile virus, and then spread to New York State generally, then spread to the eastern states, and it slowly worked its way all the way across the country. It made its way to Washington and Oregon, oh, 5 or 10 years ago now. Any question about any of that? All right, that's it for chapter 1 and chapter 10. Let me briefly start chapter 2.
You're not sharing your screen anymore.
Oh, sorry. I meant to do that when I return to the lesson. Chapter 2, Chemistry. The major goals is what I'm showing here in a rough outline. Know the terms. What's an organic compound, an inorganic compound? What's a hydrocarbon? What are polar and nonpolar molecules? What are hydrogen bonds? What's an acid? What's a base? And what is the pH? You should also, with respect to the four major biological molecules, be able to A, describe the relationship and the characteristics of monomers and polymers. 2B, describe the composition, structure, and function of the four major biological molecules. And then 4C, be able to give examples of each of the four major biological molecules. The four major biological molecules are molecules you only find in living things or something that was once alive, like if you find it in a fossil, because the fossil was once alive. If you find it in the soil or soil, it's because something living made it and then secreted into the soil. Anyways, what are the four major biological molecules? The four molecules of life. You can get three of them on any box of cereal or anything that's labeled with food. So what are three of them? Come on.
Carbs, lipids, and proteins.
Carbs, lipids, carbohydrates, lipids, and proteins are three of them. And the fourth biological molecule is nucleic acids. All right. In chapter 2, you should also know the scale of things. Let me blow this up. Where we have a human, one of the largest things we'll talk about. There's a chicken egg. The largest cell all of you have ever seen is the yolk of the chicken egg. And there's a frog egg, one of the smallest cells that can be seen with the human eye. And then there's eukaryote cells, most of them being smaller than a frog egg, and obviously it's much smaller than a yolk of a chicken egg. And then smaller than a eukaryote is a bacteria. Smaller than that is the viruses. Smaller than that, a complex molecule such as a protein. And then a smaller than that would be a small molecule like water. And then smaller than that, atoms. You do not need to know the exact sizes of things here, but you should know the rank order. Meaning a virus is larger than a protein, and a virus is smaller than a bacteria cell. Any question about any of that? I'll point out that this graph has an error in the size here. That is 1 micrometer. That is not 1 micrometer. That should be 1 millimeter. All right. What are organic compounds? They are typically complex structurally compared to an inorganic compound. They typically have carbon in them. Carbon is important because carbon combined to 4 other atoms, and they are mostly held together by covalent chemical bonds. An inorganic compound typically is much simpler than an organic compound. Typically, it lacks carbon. Doesn't always, but typically it does lack carbon. They can be, they generally are required by living organisms for the organism to grow, repair itself, maintain itself, and reproduce. Some examples of an organic chemical compound include oxygen, water, and carbon dioxide. You'll note the carbon dioxide, which is one of the examples of an organic compound, does contain carbon. Water is one of the most important and abundant molecules in life. So the most important, or one of the most important and the most abundant water molecule in life is actually an inorganic compound, water. Water is a polar molecule. What we mean by a polar molecule is a molecule that has a charge on one end and another charge on another end. This is a partial charge, meaning the oxygen has a partial negative charge that is not a complete -1 charge. And then the hydrogen, the same thing, it's a partial charge on it, not a +1 charge. The reason for the polar molecule is because oxygen pulls on the electrons in this covalent bond holding the hydrogen and the oxygen together. Oxygen pulls on the electrons greater than hydrogen, so the electrons spend more of their time with oxygen than they do with hydrogen, giving hydrogen a partial positive charge and oxygen a partial negative charge. The polarity of water allows water to form hydrogen bonds with other polar molecule. For example, water can form hydrogen bond between different water molecules. So there's one water molecule, and there's another water molecule. The positive hydrogen here will be attracted chemically, electromagnetically, I guess I'm saying, to the negative, partially negative oxygen here. And this we call a hydrogen bond. So water has two chemical bonds in it. It has a hydrogen bond between two liquid water molecules, and it has a polar covalent bond within a water molecule, meaning holding the hydrogen and the oxygen together is a polar covalent bond, which you only need to know is a covalent chemical bond. Any question about any of that? All right, Totoro doesn't call it a partial charge. He calls it a slight charge. Oxygen has a slight charge, hydrogen has a slight positive charge. It is not a complete charge, a -1 or a +1. Any question about any of that? Water is a good solvent because water can dissolve all polar molecules and all ions, meaning a molecule that has an ionic chemical bond can be dissolved by water. For example, sodium chloride, table salt, will be a crystal like this. Let me blow it up. Where the sodium has a +1 charge and the chloride has a -1 charge. So these are complete charges. And that will create an electromagnetic attraction between the sodium and the chloride. And that will form a ionic chemical bond between the positive and the negative ions will be, come on, mouse. The positive will be attracted to the negative, and that we call an ionic chemical bond. That can give rise to salt, where all the salt is in here, the sodium chloride being held together by a ionic chemical bond. Water has a partial negative charge, so the water molecules will come up to the sodium ion and then surround it and then pull it out of the crystal. And then the oxygen will be facing the partial negative charge, will be facing the complete +1 charge of the sodium. And the same thing with the chloride ion. Water will come up to it, and the hydrogen portion of the water will be against the water, and then the partial positive will be up against the complete -1 charge of the chloride, and that will pull the chloride ion out of the crystal and then bring it in solution of the salt water. Any questions about water being a good solvent? So polar substances and ions can dissociate in water, forming solute. So anything that is dissolved in the water is called a solute. So the chloride ion in the sodium ion is the solute, or the salt is the solute, and then the water is the solvent. Any question about any of that? All right, if not, I'm going to end it here, and I will continue with that in our next lesson. Any questions before I leave? I'll see you in the lab uh, at about 6:31. I'll give you a 10-minute break. All right. Bye.